A very warm good morning to all of you. So, in continuation with the series of lecture which we have planned in this short-term training program. So, today I am going to take you to the world of proteomics. So, this is very interesting also. So, if you see the title says Proteomics, a challenging but powerful tool for crop improvement, right? So, if you see this slide, I have started this slide with the world of omics. So, this omic term has been coined from the word Om. So, every morning in our house we used to chant Om word. Om means omnipotent, means present everywhere. It means when you add omics to any of these tools, it means you are going to quantify all that particular macromolecules which is present in your specific tissue at particular event of time. For example, we have a genome. Genome means you are going to identify and characterize all the genes which are present in your particular tissue at particular event of time. Then you can use the term genome. Similarly, proteome means you are going to quantify, you are going to identify all the proteins which are present in that specific tissue at particular time. Then you can use the term proteome. Many times I have seen people, they'll use this proteome for 2, 3, 4, 5, 10 samples. No. The real objective of using the word proteome is protein plus omics, means whatever protein are present, wherever it are present, that you are going to identify and quantify. Similarly, you have a metabolome, means you are going to identify all the metabolites which are present in a specific tissue or in a specific closed arena, right? And if you see the classification, so we have then different sub-classifications of all these omics which was discovered and characterized maybe 4 or 5 years back, right? Like now you have whole genome bisulfite sequencing, you have a miRNOM, means you are going to identify all the microRNA which are present in a specific tissue, right? Similarly, in metabolome, you have different subsets, like you have a lipidome where you are going to identify all the lipids. You have an ionome where you are going to identify all the 92 elements which are present in a specific tissues. It means when you are going to identify something in a high throughput manner in an exhaustive way, that signifies or depict the omics, right? If you see the classification of proteome here, so in proteome, I think gel based proteome we all know, and in our lab we all are using, like SDS Phase or 2D Phase. So most of the time we are using in our lab. But now from last 2 years, gel free proteome is something a buzzing word. So people, they rely or they are using this gel free proteome in an exhaustive manner because of certain advantage. So proteomics has been broadly classified into gel based and gel free method. And then metabolome, so other classifications I have given, like you have a traditional method, you have a non-targeted, and then you have a next generation technologies method. And when you clubbed all these methods, so all these methods in a clubbed way decides the phenome or basically the visual observation or phenotype of the plants. Means whatever trait you discuss, so that particular specific trait is influenced by one or the other omics platform, right? So all these subsets interact together and design or decides the phenome, right? Coming on to proteome, so Mark Wilkins, he was the first person who coined this term proteome, and he signifies that when you are going to identify all the proteins which are present in a specific tissues at particular event, then you can use the term proteome. For example, suppose if I have a root tissue, root tip, and I want to know all the proteins, whether it is known or not known, whether it is unknown. So if I want to identify, if I want to quantify all those proteins in a specific tissue at particular event of time, then I can use the term proteomics, right? So basically all these omics are done in a high throughput manner, and most of the part, almost more than 75% of the steps are machine based. What we are doing is, we are preparing the sample, we are just doing the fractionation, we are enhancing the quality, we are enriching the samples, and then once you feed into the instrument, your job is done. Then all the instruments which are available and used in case of proteomics are well equipped to characterize, to run, to characterize, and to interpret, and it will give you the conclusion also. Right? And if you see some of the uses of proteomes, so here if you see I have started with 2D. So that upper one, this is a 2D gel. Then if you see downstairs, so this shows some of the Western blot, which is also one of the prominent tool used in case of proteomics. Now if you see recently a paper has come out in Nature where people they have discovered protein targeted based breeding, which is also called as precision breeding program, where they are going to use a specific antibody to screen thousands and thousands of samples in one go using the Western blot method. Similarly, we have the mass spectroscopy, which is the backbone of proteomics. For any identification, you have to use this MS-based methods. And then these are some of the dyes gel. Dyes is basically a differential gel electrophoresis gel, that last one. So there we are going to label different proteins, and then you are going to club it and run it on the gel. Right? And some of the uses, if you see, of the proteome is, so most of the characterization or most of the use which people they have done is in the area of abiotic stress tolerance. If you sit on the internet and if you find the papers, good papers, so almost more than 80% of the papers, this proteome has been dedicated to abiotic stress tolerance area, where people they have used this to decode, to explore, to decipher different mechanisms which are used or which is operating to modulate the tolerance of any plant. Then next is biotic stress. So this proteome is very popularly used to validate the findings in case of biotic stress tolerance. The third area is crop quality. So isolation of these storage proteins, their characterization, accumulation pattern, quality of these storage protein, quality of the grains, all these features people they rely on proteome. Now if I ask question, why people rely on proteome more than the gene or the midway technology? It's only because it is the proteome which is present at the site of action. If you have certain trait, if you have certain feature, it means some reaction is going on because of which you are getting a specific trait. And if that reaction is going on, it means you need some enzyme, and all enzymes are proteinaceous in nature. Right? And the last one is growth response. So this is also one area which has exhaustively utilized the tool of proteome to decode the mode of action of different growth hormones, phytohormones, not only in plant, even in human being also. Proteomes are very popular tool to be used in medical sciences. And let me tell you, some of the tools which we are using in proteome in agriculture science, that has been borrowed from the medical science. So in subsequent slide, I will tell you how we have borrowed that one. So now coming on to evolution of proteome. So I think till date, if I ask you different generations of proteome, so we know only one generation, that is something which is based on gel and SDS phase or 2D. But then we have very specific characterization of different generations of proteome. So first generation is when people they were doing research based on qubit. Qubit means you are going to get a color, get the OD value, and then you interpret the results. And if you go 40, 50 years back, this first generation tool was the most popular. People they were happy seeing the colors, feeling that reaction is going on. Then comes the second generation. Second generation means in gel activity. People they started showing the reaction on the gel. For example, enzymatic reaction or electrophoretic profiling of enzyme or isoenzyme profiling. So that was second generation when people they started doing all these reactions on the gel, and this was very popular. Then comes the third generation where people they started doing 2D phase, one-dimensional SDS phase and all those things. The idea was all these discoveries were based on the limitations of the earlier technique. Right? So 2D was discovered because in a gel you can show the activity reactions, but if I want to profile the protein, I cannot. Or if I want to get a fingerprint of the protein, I cannot. Then they discovered this 1D and 2D phase technique. 2D is two-dimensional polyacrylamide gel electrophoresis, where you are going to separate your protein using two different methods clubbed together. right? The idea was we are going to put lot of protein spots, or we are going to accumulate lot of proteins on the gel. So in case of one dimensional paste, this was one of the problem, that only limited bands were visible on the gel. So that limitation was rectified by two-dimensional paste because of protein spots. So lot of protein spots were visible or accommodated on the gel. Then comes fourth generation proteomic tool, and fourth generation was like a revolution in the area of proteomics. That is mass spectroscopy based technique, where people, they thought that let us shed this gel business. Directly take a tip, extract the protein, run it on the gel and run it on the machine, and then find out what are the proteins which are present in that. And from last 8 or 10 years, MS has ruled the proteomics world like anything. If I have to find out the protein which is present in leaf, I will take the leaf, I will extract the protein, I will feed into the machine. It will tell me based on the database which is available publicly that what are the proteins which are present in my leaves. All right. Then people, they started characterizing this MS based method also, and now we are in an era where we have two very popular methods under fourth generation proteomic tool. One is called as label based proteomics and other is label free proteomics. Label based, for example, iTrack. iTrack is basically isobaric tag for relative and absolute quantitation of protein. iTrack, isobaric tag for relative and absolute quantitation of protein. So this is label based method. Isobaric tag for relative and absolute quantitation of protein. And then we have label free method. Label free method means directly you are going to sequence your protein. So whatever sequencing result you are going to get, that you are going to align, and then you are going to match it with the reference library which is present, and then that will tell you which protein is present in your sample. Now the advantage is, in all gel based method or maybe simple MS based method, you can find out only those protein which are known, which is characterized, reported, present in your library. But this label free or label based method, this will tell you all the novel or hypothetical or known protein which is present in your sample. Means the protein which you don't know, that also you can identify. Now see the beauty or the power of this particular instrument. Suppose in few of the castor crop, I think male sterility is one of the problem. So in Gujarat, this was one of the biggest problem, and then people, they were not knowing why it happens, what are the proteins which are instrumental. So we worked with some of the institutes over there, we did this label based method, and first time we find out 2 or 3 hypothetical protein which were basically proteases. So these proteins were the culprit for this particular sterility. Now if I use any other tool, it's very difficult to find out because we don't have any reference. We don't know which because when you isolate, when you run, everything will come, but I don't have identity how to validate which protein is this. But then once you get the peptide sequence using these methods, so easily you can match out, you can make an antibody out of that, you can screen, and you can easily isolate also this specific protein, and this is something like novel work. Same thing we did in case of wheat. I will tell you that Rubisco activase, those who are physiologists, they might be knowing. Rubisco activase is a catalytic chaperone which converts inactive form of Rubisco into active form. So by default in plants, Rubisco is present in inactive form. So this activase, it will come, it will convert inactive into active, and then photosynthesis starts. But nature, they are very clever. They have designed this Rubisco activase enzyme in such a way that it is highly heat labile. Heat labile means even if there is a fluctuation of 2 or 3 degrees Celsius in temperature, this will denaturate. It means once Rubisco activase is denaturate, so Rubisco will be in inactive form, means photosynthetic rate will slowly dips down. It means carbon fixation is hampered, right? In case of wheat, when we started working using these tools, only one partial sequence of Rubisco activase was available on public domain. So we started characterizing, we started exploring, and then we got project also. And then let me tell you, using these tools, first proteomic tools, that is iTrack, I could able to find out almost more than 14 or 16 Rubisco activases. Then using the transcriptomic tools, we validated all those Rubisco activases, and out of that 8 we cloned, characterized, submitted in NCBI GenBank, and then out of 8, 1 we selected, we manipulated using the protein engineering tools, and then now we have a Rubisco, mutated Rubisco activase which can tolerate temperature up to 42 degrees Celsius. Now see the advantage of this. If I would have not used these tools, you please tell me any other tool through which I can find out in case of wheat, which has a very complex genome, that how many Rubisco activases are there, and if it is there, what is the identity of that, how to find out this. But it was possible, all this story was feasible only because I used these omics tools. Right? So this is the potential, this is the power of these tools, and those who are doing research, wet lab is one part, but then you should equip yourself to inquire or to query into the areas of omics, so that at least you should be filled with all these informations, and then you can use your brain to find out in which area, in what way we can venture. Right? So all ideas come once your kitty is filled with information. So here you can see I have already explained this label and label free method. So our lab is equipped in such a way that we are using almost all these tools. Some of these tools we are using at our own lab, and few of the instrument, especially for label and label free method, it's very costly. So we are availing this facility from common facility at JNU or from outsourcing to companies people, because the biggest lacuna with these instruments is that these are very costly. This is the only limitations attached with these instruments. Costly means cost is in crores. This is one of the reasons why proteomics are not so much popular as transcriptomics or as metabolomics, because we can plan, we can think, but when you have to access that particular instrument, it's very difficult. So this is one of the major reason behind the failure of this particular technique, and now we are in an era where people, they doesn't like to use proteomics, even though this is a very beautiful tool. When you see your dye gel, when I'll show you some of the results, when you see your 2D page gel which you have run, that gives you the maximum satisfaction. So these are some of the techniques. So I have just clubbed here like two-dimensional electrophoresis. Nowadays also people, they are using it in an exhaustive way, where first dimension you are going to separate your protein based on the isoelectric point, and second dimension you are going to separate based on the molecular weight. Similarly, you have a ICAT, that is isotope coded affinity tag. So some 5 years back, this was one of very popular technique which people, they are using. But because of use of radioactivity, and then when Bark come into convention show, they stopped this. They told only accredited lab can use this particular technique. So this technique was later on obsolete. Then you have a MS based technique where you are going to run, and then you are going to match your mass to charge ratio table onto the database, public database, and then that will give you the peptide sequence or the identity of the protein which you are going to use. And then these are some of the fingerprintings. Now important is the databases which I have listed here. This is another lacuna associated with proteomics. The problem is I have data with me, but I don't know how to characterize this. And even if I characterize, I feed it onto some of the databases, they will ask you first pay the money, then you access my database. Like suppose NCBI starts asking you, first you pay, then we'll allow you to go for BLAST10. So it will be devastating for the research. So that problem is happening with the proteomics. You run, you have a table, you have a data with you, but we cannot characterize because freely accessible database is limited. This is another problem. So here if you see some of the databases which people, they are very popularly used, like PRIDE, Proteomic Identification Database. This is a database which helps you in decoding the MS based data. You feed it, it will tell you what are the proteins based on the peptide sequence. So you have a Proteomic database, that is protein database access. You have a PPDB, that is plant proteome databases. Till date, it has been designed for Arabidopsis and maize only. Then you have open proteomic databases. It allows whatever data you have, you feed it, and then it will give you the identity of the protein which is present in your sample. I think there is problem with this. So coming on to SDS Phase, I think this is a technique which almost most of you might have used in one or the other part of your career, right? So the principle is very simple, that you are going to use anionic detergent which basically linearize the protein, and then you are going to separate your protein based on the molecular weight. But one thing which you might be not knowing is this is also called as 3-gel system, right? This is also called as 3-gel system, or it is also called as 3 pH system, because we are using 3 different pH for separating the protein. And right hand side, if you see, I have listed few of the uses of this particular technique. So SDS Phase is one of the most reliable technique in the area of proteomics. For any validation, for any of these tools which you are going to use, so SDS is like a backbone. Right? Some of the uses, like you can use it to find out the molecular weight of any unknown protein. You run it, and then you can match with the marker, and then you can say that what is the molecular weight. It is also very popularly used for protein profiling. Many times people from horticulture, genetics, they used to come, they want to know the protein pattern profiling of a specific tissue. So you run it, and easily you can compare across the genotypes. What is the variation? So whether any new protein is present in my sample or not, all those informations easily you can make out by running it on SDS Phase. Similarly, for determining the integrity of the protein. Now suppose I am working in the area of quality. You run it on the gel, and based on the intactness of your band, easily I can comment whether the protein quality in my sample is good or not. The protein, the storage protein which is present, whether that quality is good or not, easily I can comment by running it on the SDS Phase. Similarly, for protein quantitation, you run it on the gel with some standards whose concentration might be, I might be knowing, then easily you can tell what is the concentration of my protein which I have loaded onto the gel. So for the quantitation also, this is one of the best method. And for 2D, so this is one of the dimension of 2-dimensional phase electrophoresis. And this is very popular, easy to use, and then it gives lots of information not only to the biochemist, across the science. So whether it is a breeder, whether horticulturist, pathologist, people, they are using it in one or the other way, for screening, for validating a protein through Western blotting, for identification of a protein, for characterization of a protein. So n number of uses this SDS Phase has. Coming on to 2D phase. So the basic principle is very simple. You are going to separate the protein first based on the isoelectric point, and second you are going to separate based on the SDS Phase. The only advantage with 2D phase over the SDS Phase is, here you can accommodate more number of proteins as compared to SDS Phase. Second is, here resolution is very high as compared to SDS Phase. Third, easily you can pick protein spots from here, you can subject it to MALDI-TOF, so that easily you can identify the protein at this step itself. This is another advantage, and till date also this is one of the most favorite method which most of the researchers they prefer to go for, because you don't have to run between the pillar and post. SDS Phase you can do, isoelectric focusing that you can do in your lab, and then identification. So MALDI-TOF MS is now easily accessible, almost all the divisions they have. So you run, you have lot number of protein spots here. Based on the characterization, you pick spots, go for tryptic digestion, and run it on the MALDI-TOF MS. You will get the identity of that particular protein. Coming on to subsection of proteomics. So proteomics now you know. Now there are different subsections of proteomics. For example, quantitative proteomics. So all these eye track, label, label-free method, all these are quantitative proteomics. As the term suggests, quantitative means quantity. You are going to identify, you are going to characterize, you are going to count the quantity of this particular protein. Next is phosphoproteomics. So phosphorylation, you might be knowing, is one of the very potent regulatory method in case of any enzymatic reaction. So most of the important key metabolic pathway associated enzymes are phosphorylated or phosphorylation based regulation is there. So this phosphoproteomics will empower you to find out all those phosphorylated enzymes which are present in your sample. Coming on to proteogenomics. So proteogenomics empower you to find out all the proteins or characterize the protein based on the protein level or gene level or transcriptome level. So you can connect all these three using the proteogenomics tool. Last is glycoproteomics. So all these membranes, they have a glycosylated different forms of carbohydrate present over there, different modifications are there which plays very important role in sensing or signaling. So if my objective is to find out all those proteins by using glycoproteomics, easily I can find out. And now the buzzing word is cis proteome. We are going outside, we are eating a lot of things. Now cold is there, a lot of environmental variations are there. So all these protein respond to these environmental variations. So all these environmental variations are called as exposomes, and the changes or the variations or the pattern change in the protein going inside. So scientists, they have validated that this cysteine shows maximum change in response to environmental factors. And then they coined the term cis proteome, and which has now been very prominently used in medical science to predict any disease or to predict the mood or predict the behavior of a person well ahead of time. Cis proteome. So some of the softwares which you should know, that these are the name of the softwares and for which purpose we can use, like XPacy tool. I think most of you might have used this. XPacy is a web page where you will get thousands of softwares which are available, and most of them are freely available. So you can feed your data and it will characterize for n number of things. Then you have a Proteome. This is basically used for protein structure evaluation. So if you have amino acid sequence, you feed it, it will model the protein and easily you can characterize. You have a ProRaginine. So this is for globular protein evaluation. Then you have a Mascot. Mascot is like a primary library or database for MS based data. If you have a mass to charge table, you just copy it into the Mascot. Mascot will tell you which protein it is. So Mascot is a database very commonly used for MS based data. Then you have a Scaffold, which is basically used for MS2 based data. Then you have a Decon MSN, which is used to create a spectrum profile, means protein spectrum, the fingerprint of protein which you are going to create. So this software will help you to create that spectrum. So like this, n number of databases and softwares are there. The only problem is most of those softwares are paid one. You have to pay money to use. There is one more software which is called as Fire 2.0. The beauty is all mutation related work that you can find out, all structure related work. You just feed amino acid, it will tell you the structure of the protein. It will tell you the hot spots which are present in the protein. It will tell you what is the modifications in that particular protein. It will tell you where is the active site of that particular protein. All sort of characterization you can easily do at one place, that is Fire 2.0. Now coming on to the proteome and its uses. As I told, basics I have conveyed. Let me validate my word with my own results. So seeing is believing. If you see the results, then you will have a trust that definitely even if he can, why not I? So some of the gel based proteome, as I told, my expertise is in the area of stress proteogenomics and my crop is wheat. So most of my results will be in the area of wheat, but then these results are easily replicated in other crops also. For example, this is differential protein profiling in case of wheat. So when we planned this, our objective was to find out all the differentially expressed protein which are present in wheat and which expresses in response to heat stress treatment. Now if you see, first gel is the control, second is the heat shock treated. So this we did almost 2012. or 11. That time it was new for us, but this was done at our lab. And if you see, we could able to find out couple of differentially expressed proteins, and then we pick few of them and we subjected to MALDI. Now these are the list of proteins which we could able to predict based on MALDI of MS. This is the table of mass to charge ratio. This is the peptide mass fingerprinting. So you have to feed this table into the database, and it will tell you the identity of the specific protein. It will not only tell you the identity, but it will tell you the closely related protein also. Now beauty is, once I get this particular table, top one, so easily I can differentiate all these protein based on the functions. And if I differentiate this, if you go through some of the papers, they will tell you that I identified this much protein for defense, this much protein for nutrition, this much protein for photosynthesis. It is these proteins only. So once I get this data, I can differentiate it based on the functions, and then now I have a huge data with me. So these are some of the list of the proteins. So these are some of the key proteins. Especially first time our lab reported that oxygen evolving enhancer protein is one of the most abundant protein which is expressed in response to heat stress treatment. First time we claimed this and we proved this. And one of our MSc student, Ansif, he completed his MSc also on that particular protein. He cloned the gene, he did expression analysis, Western blotting, characterization, all sort of things. So now, if I don't know to use these tools, it's very difficult to find out, to come to a specific conclusion. If you see the list here, these are some of the proteins which are very important, like some of the hypothetical protein, ATP synthase, small heat shock protein. Now let me tell you, we recommended to Division of Genetics that a small HSP can be used as one of the potent marker for screening wheat for thermotolerance. And downstairs, so now relative fold expression, just few minutes back we were discussing. Now this is not only a term which is used for gene expression. In protein also now we are using relative fold expression, and this expression is based on the volume or the pixelation or the area of the spots which is present in control over the volume or the pixelation or the area which is present in heat shock treated. So when you divide this, the ratio which you are going to get, that signifies the fold expression of that specific protein. Here we have done for some. And whatever I am presenting here, all these are published ones. So easily you can go back and you can read also. So these are some of the relative fold expression which we did using, just I have shown one 2D page, but we run almost more than 100 2D pages. So these are some of the expressions which easily you can identify, you can characterize, and then you can club at one point. And this will help you in remodeling the pathway. Once you have identified the specific protein, right? Like I have identified some 50 or 60 potential protein which plays very important role in modulating the thermotolerance in case of wheat. Now how to validate this at gene level? So that technique I have already explained you, real-time PCR. So gene sequence is available, you just find out the complementary gene which is present in NCBI. You design the primer, go for the expression analysis in the same sample to find out whether the expression of gene matches with the expression of the protein or not. Now you can see the result here. We selected some of the proteins, like first table shows the heat shock proteins. So like HSP90, this is a heavy molecular weight heat shock protein. You have HSP70, this has a dual role for protein folding as well as tolerance. You have a HSP26 or 17, so this plays role in folding of protein as well as defense. Like this, you have antioxidant enzymes, you have signaling molecules like rubisco activase, calcium dependent protein kinases, calmodulin, all these are signaling molecules. You have a pathway associated genes. We did expression analysis and we got wonderful graph or cluster analysis of this expression with the protein, and it was most of the time it was 99% correlated. The story doesn't stop here. Once I validate the transcripts, then you have to validate at protein level also. So for protein level, the best tool is Western blot. Now if you see the Western blot result here, So we designed the antibodies, we synthesized the antibodies, and we did Western blot in an exhaustive manner. And then now we are in a position to say that the expression of gene increases this much time. Because of this, the protein expression was this much fold. Because of this, the accumulation of protein was this much. So we have done Western blotting in exhaustive manner for different proteins which we identified using the 2D phase. One of the result is this. So here you can see the accumulation pattern of HSP70 or calcium dependent protein kinases or HSP90, and we proved that when you give heat stress, all the accumulation of these heat shock protein increases. And the problem is, if duration increases, so accumulation will slowly go on decreasing and it will also denaturate itself. And then plant will decide that now there is no way out, let us kill the cell. This is one experiment which we did. This is in response to hormonal treatment. So we selected different concentrations of GA and then abscisic acid, and then we thought, let us see what are the hormone responsive proteins which are expressing. And if you see this graph and compare with the control, so with response to heat stress treatment and with response to these hormones, so easily you can see we characterize this also, and we could able to find out almost 9 to 10 proteins which were novel one expressing in response to this hormonal treatment under heat stress. And then we cloned few of genes also, and then we characterize, we got very good publication also, and we are now exploring to see the use of these as a markers. Pollen, I will tell you wheat pollen. So 2014 there was a notion worldwide. So the notion was that when a pollen is detached, so pollen does not have any system which can express the heat shock protein or which can protect the pollen from the heat stress. So first time using the tools of omics, we validated, and through wet lab we proved that no, even if pollen detaches, it has a potential machinery to express heat shock protein. And it not only express heat shock protein, but heat shock protein also protects the pollen from the external abiotic stress, especially heat stress. And first time we proved that no, heat shock protein is expressing even in detached pollen through real time, through Western blot and other tools. It is first time we identified 2014 itself that wheat pollen has apostules or pores on the surface, and these pores are plugged with a starch. If it is plugged with a starch, pollen will be viable. If a starch come out, that pollen will be flaccid and it will not able to grow. It means no fertilization. No fertilization means no seeds. And we characterized, we proved this using scanning electron microscopy also. So all this happens because I know the tools, which tool at what place I have to use. So that was gel based proteome. Now let's have a look on the gel free proteome, where no need to make a gel, directly we can quantify the protein. And one difference between gel based and gel free method is, gel based method you can identify 40 to 50 protein at max. In case of gel free proteome, you can identify 4000 to 5000 protein, and if you are using some labeling based method, you are going to identify 45,000, 55,000 protein in your tissue. Now you compare 55,000 protein with 40 or 60 protein in same tissue. How much data you are going to generate using the label free method or label based method or the 4th generation proteomic tools. And this is a power, once you identify, once you have that much protein with you, easily you can simulate those proteins, you can develop an interactome of those proteins, you can simulate the pathway itself. One work which we are doing along with Dr. Suneha Goswami is, we are remodeling the starch biosynthesis pathway itself. So as a student, if I ask you, there are 4 or 5 enzymes which we know from last 30 years. But first time using the omics tool, we could able to figure out 67 novel gene and protein which are key member of a starch biosynthesis pathway, and the world doesn't know about this. These all feasible because we know these tools. So this is a basic graph flow chart which shows some of the technique which you can use for the gel-free proteomics, like you can go for the orbit trap. Orbit trap is basically a gel-free method and is a nano LC MSMS. So orbit trap, the difference between orbit trap and other higher end instrument is orbit trap is cheaper one. Within 12,000, 10,000 rupees you can run your sample, you can characterize. But other methods are little bit costly till date. So you have a MALDI, so you can collect a sample, you can go for one dimensional page, you can go for two dimensional page, you can feed it into the MALDI TOF MS to identify the protein. If you don't want to go via that route directly, you can extract the sample and run it in the orbit trap. That will tell you the peptide sequence. You just, based on the peptide, you identify the protein. Even if you don't want this, you isolate the protein, go for fractionation, go for triptych digestion, feed it into the labeling machine, and then you will get the identity of the protein with high resolution. So these are three different flow charts or the workflow charts which basically people they are using, those who are working in the area of proteomics. Global proteome profiling. So this is one very common term when you sit and when you start searching. So this will come every second slide. So this is basically a tool where you are going to identify thousand and thousand of protein using the power of MS, mass spectroscopy. And here you can access even difference between the sample that easily you can make out by running two different samples simultaneously. Even you can discover some of the isoforms of this protein also, and then modifications also easily you can characterize, you can identify, and you can conclude. This is a result of orbit trap. So when we started this, our idea was to find out some of the proteins which are expressing in response to heat stress, and we did orbit trap, which is basically nano LC MSMS. If you see right hand side, so based on this, we could able to figure out couple of proteins which are very important one, playing very important role in heat stress tolerance in case of wheat. For example, heat shock protein 70, which has a dual role. It helps in protein folding also, and then it helps in protection also. You have ATP synthase, you have some of the predicted protein which are pathway associated, we have some of the predicted protein which are photosystem 2 associated, some of the small heat shock proteins, and then signaling molecules. So these were the prominent proteins which were observed to be expressed in response to heat stress in case of wheat. So we thought that the problem with this technique was, so we could able to identify some 80 to 90 proteins only. Then I thought it's not possible that the particular tissue has only 90 protein. It means there are many unknown protein also which we should find out. Then we started doing the gel-free labeling method, as I told, which is iTrack, and this is one of the best 4th generation proteomic tool. The full form of iTrack is isobaric tag for relative and absolute quantitation of protein. So this is 4th generation proteomic tool, and this tool we have borrowed from the medical science. So in medical science, people they are using this for cancer study, for different diseases characterization. We borrowed this, and now this is spreading like anything in the area of agriculture science. The advantage of this iTrack is, in one go in a specific tissue, I can find out thousand and thousand of proteins with high resolution. High resolution means if I go via reverse genetics, easily I can figure out the gene and I can clone that gene also. So in case of iTrack, if you see, so this has a 3 different module. One is called as reporter ion, other is called as a balancer group, and then you have amino specific peptide reactive group. So this peptide reactive group, basically this forms a bond with your protein, and then this balancer group, it creates equivalent molecular weight. If you see the right hand side, so the workflow chart is very simple. You have different samples, you are going to do triptych digestion, and once you have done triptych digestion, different dyes are available. iTrack means iTrack dye is there. So different dyes are there with different molecular weight. You are going to label your different samples with these dyes, and once you have labeled, mix it and then go for run. Once run is complete, you will get a specific pattern of sequence. You feed this pattern into the public reference database, and once you feed this, that database will give you the list of all the known as well as unknown as well as hypothetical protein which is present in your sample. Now see the difference. If you are doing gel based, so you are going to identify 5 to 10 or maybe 15 protein. If you are doing gel free method, some of the initial method you can identify 80 to 100 protein. But if I am doing eye track, you can identify more than 5,000 to 6,000 protein. So you can see this is one of the results. So I am struggling hard to operate this. So this is one of the results. First time we did in almost 2015 when people they were not knowing much about the eye track in agriculture science, and we could able to identify almost 5,500 protein with high resolution. And we not only identified, when we started characterizing the data, so you can see right hand side. So these are list of some of the proteins which shows overexpression in case of thermotolerant cultivar, but down regulation in case of susceptible cultivar. Now please tell me name of any tool which can tell you in one go that this much protein is overexpressing in tolerant, but this much protein is down regulating in susceptible. We don't have any tool like that. So this is the only tool which will help you, empower you to find out the upregulated protein, same protein which is upregulated in tolerant but downregulated in susceptible. And these are some of the other localization. And then once you have a eye track data, n number of tools are available which you can use to characterize the specific protein which you have identified, their function, their structure, their localization, their behavior in case of tolerant and susceptible. All those things you can do just by doing dry lab work. And once you identify the protein, so as in my initial slides I told you, then you have opportunity to execute n number of wet lab work also, like some of the potential protein. If you want, you can do the accumulation pattern profiling using Western blotting. If you want, you can go reverse genetics to clone the specific gene which is responsible for the expression of that protein. You can do the cluster mapping to find out the correlation between the expression of the gene and the accumulation of the protein. You can do localization study of that particular protein. You can design the structure and then you can do the activity assay of that specific protein. So n number of wet lab characterization you can do, and you can generate ample of results to get a conclusive model like this, like we did transcriptome as well as eye track, and then whatever expression and activity assay result we got. So we designed or simulate a model that under heat stress how the starch grain or the structure of a starch grain influence, and what are the partners or what are the enzymes which is influenced by the heat stress, and how the expression and activity of this enzyme varies under terminal heat. So we designed a model itself, and this we published in Functional and Integrated Genomics. So like this, in your area of research, once you get so much data, so easily you can simulate a model. Not only model, you can devise an interactome itself, like the protein which you have identified, whether that protein interact with how many other proteins, in what way, what is the impact of interaction, why this interaction is happening. All sort of questions you can answer using these label-free methods. Now one question which is very common, and most of the time people they used to ask that what is the basic difference between label based method and label free method. So if you see some of the categories which I have written here, like machine time, so in case of label free method, that takes more time for the run as compared to labeled method. And in case of labeled, it's always easy to catalog, to align the protein. But in case of label free, since directly you are going to sequence, sometimes it's very difficult to align that protein. Then if you see the wet lab complexity, so definitely this label free method, it's easy because directly you are going to run it. But in case of labeled, so you have to label it after tryptic digestion, then you have to club it, and then you are going to feed this into the instrument. And other than this, so design and other things, so overall the view is that labeled method is better than the label free method. This is the conclusive out of comparison of these two techniques. Just two slides I will show you in the area. So last year we got one project from ICR which is on pearl millet. So in case of pearl millet grain, rancidity is one of the biggest problem in pearl millet powder. So when you crush this, so different enzymes are there which just degrade the lipids, and then because of the free fatty acid chain, so you have a rancid or off odor in pearl millet powder. So I did some of the SDS Phase to find out the protein pattern profile of some of the storage protein which is present in pearl millet grain. So here so it's very difficult. So here you can see we selected 4 prominent genotypes of pearl millet, like white, purple, Dhan Shakti, which is recently released by ICRISAT, which is fortified with zinc, and then decorticated Dhan Shakti. And you can see the difference in the pattern of the protein band which is appear on the gel. So easily you can differentiate between these 4 genotypes. And not only this, so we make the powder of this, we isolated the soluble fraction of this storage protein, and then we run it on 0 days, and then the powder which was kept for 20 days to see the change in the protein pattern. So you can see in the sample in case of 20 days, so almost many of the protein bands disappeared, right. And then we did 2D Phase also, because our objective was to find out these specific enzymes. So we did 2D Phase also. So recently, so we got result of this. So here you can see the 2D Phase of grain of this spike and then leaf of Pusa 1201, which is one of the popular pearl millet genotype cultivated in IRI. So now we are doing characterization with this. So these are some of the publications. So you can go and you can type the title, so easily you can retrieve these papers, and whatever I have presented, so all these are published one and available on the public domain. So easily you can go through these papers. So now I think one more slide should be there. Yes, future prospects. So now whatever I explained, so there is a need to take this into the future pathway. So like there is a need to explore the redox proteome, because redox, as you know, oxidation and reduction is the backbone of all the biochemical reactions which is going on. So there is a need to explore the redox proteome. Then there is a need to connect genome with the proteome. And then if you see, so as I told, so all these are very costly technique. So now this is time where we have to intervene, and then we should have a cost friendly or user friendly method with a cheap cost, so that anybody can think of and avail or access these techniques. And then software. So as I told, this is one of the biggest lacuna in case of proteome, that we don't have freely available softwares for characterizing the data. So there is a need to have access to freely available softwares. And then functional computing is one of the biggest problem, because these are big data, and some of these computers, they cannot take that much load. So we need to have a high computing server. This is also one of the lacuna that this proteome is not very much popular among the masses. And last is, this is a time where we have to connect this proteome with the phenome. So until unless that is a translational research, until unless something is visible, nobody will believe in these techniques. So these are the publications as I have already shown, and I would like to acknowledge Director IRI, Joint Director Research IRI, Head Division of Biochemistry, Dr. Vishwanathan, Coordinator School of Basic Sciences, Dr. Himanshu Pathak, because the result which I showed during 2012-13, so he was the PI of one of the mega project, that was NICRA project. Dr. GP Singh, so whatever wheat accessions I got for characterization, so all those accessions was given by Dr. GP Singh, Director IWBR. And then my colleague Dr. Suneya Goswami, so she was always as a co-PI in almost all the projects which we are running, all my SRF, RAs and students. And a special thanks to the funding agencies like ICR, SERB, BARC, DST, DBT and CISR. So thank you all. Now if you have any queries, any questions, I am ready to answer. Please.
Sir, as you told us, it was all depend on tools and techniques in a lab. But right now we are living in a high-tech world and have so many tools, technique. We have many things on our mobiles. So is there any way that we can make these tools more cheaper and more comfortable? And is there, I mean, role that government can play in this?
Yeah, I'll tell you. Like, suppose you have a SDS, you have done SDS Phase. Now, if I want to quantify the concentration of that particular protein band, so in mobile there are different apps, which is basically a densito analysis based apps, where you take a click, you scan your gel, and it will tell you the lane, the number of proteins which are present in your lane, and what is the concentration of that protein. So apps are available, but the thing is awareness is not there. People, they doesn't know how to use that. For example, 2D Phase. Now app is there where you can take a click of 4 different 2D gel, and you can superimpose all those treated gel over the control, and it will tell you what are the differentially expressed protein spots which are present in your gel. And it will not only tell you, it will tell you the number also, that protein spot 1 is differentially expressed, and this is the increase in the fold expression as compared to control. Regarding the government intervenes, I think my last slide, the freely accessible software, high computing server, I think these are some of the things which is not possible from the government institutions or from a specific lab. This is only by policy change only. Like in some of the states, government, they have distributed laptops. So like this, there is a need that student or maybe researcher should get license to use some of these databases. They should get some of these softwares freely, and then they should get this high computing server, not individually, but at least in a group, at least they should get, so that interest will be created. And once you will start getting the result, once you see that protein is dancing, so that will itself create awareness, and then people will be interested to use these tools. Thank you. No question?
So hope you all enjoy this talk very much. Thank you, Dr. Ranjan, for giving such an informative and the talk overloaded with recent information. So thank you, Dr. Ranjan. Now I request Dr. Anil Dahuja, Professor of Division of Biochemistry, to kindly felicitate Dr. Ranjan with a moment.
It's always honor to receive from my teacher. Thank you. Thanks a lot. Thank you, sir.
So you'll all agree it was